Good morning, everyone. Thank you so much for joining us here today. We'll get started on time. I realize that I'm surrounded by screens, so you can only see like a top third of my face, and it feels like COVID all over again. But anyways, we have an excellent lineup of speakers that cross across the research spectrum, from basic science to clinical research, presenters from various backgrounds as well. So I think we'll hear some really exciting talks. So without further ado, I'd like to introduce Dr. Rowena Wood to come up to the stage.
Okay. All right. Thank you, everyone. I'm glad to have the opportunity to speak with you this morning. So I wanted to let everyone know I don't have any disclosures for this presentation. So as everyone here is familiar, the disease cystic fibrosis is a genetic disease caused by loss of function mutations in the cystic fibrosis transmembrane conductance regulator anion channel. This leads to a loss of chloride and bicarbonate conductance in cells, and the result is that there is dehydration and pH dysregulation of epithelial surfaces. So cystic fibrosis affects multiple organ systems. So in the airway, we see failure of the mucociliary clearance apparatus. In the pancreas, we see pancreatic insufficiency and mucus plugging of pancreatic ducts. In the immune system, we also see immune dysfunction, as CFTR is also expressed in a variety of innate and adaptive immune cell types. And we also see multiple manifestations of cystic fibrosis in the gastrointestinal system. So cystic fibrosis intestinal manifestations include obstructive bowel disease. We also see dysbiosis of the gut microbiome, as well as chronic intestinal inflammation. So these aspects of CF are recapitulated in the CFTR knockout mouse model, which has these manifestations without the complications of either the airway manifestations or the pancreatic manifestations of the disease. So this image on the right here is showing you a comparison between a person that has cystic fibrosis. This is showing their intestinal crypt epithelium, and comparing this to a knockout mouse model of cystic fibrosis. You see that they share the characteristic mucus plugging of the intestinal crypt epithelium, as well as the goblet cell hyperplasia. So CFTR knockout mouse models do require dietary interventions in order to prevent lethal intestinal obstruction. So this can be achieved by either providing an osmotic laxative in their drinking water, or by providing a daily provision of a nutritionally complete liquid diet. And so the global or pan-CFTR knockout mouse model has been used to study the contribution of CFTR loss of function to intestinal dysbiosis in CF, using either the PEG laxative or the liquid dietary regimens, as you can see from this list down here, and looking at dysbiosis in a variety of intestinal segments, so small bowel, large bowel, as well as fecal microbiome, most of which use the 16S bacterial ribosomal RNA in order to study the composition of the microbiome. I wanted to draw your attention, though, to this reference on the bottom here, which is a recent study performed by Skoll et al., in which they looked at a tissue-specific contribution of loss of CFTR on the development of intestinal dysbiosis. So in this study, they utilized wild-type CFTR mice and also a pan-CFTR knockout mouse model, and compared these with myeloid-specific CFTR knockout mice, so in which CFTR was knocked out in granulocyte cells as well as monocytic cells, as well as a neutrophil and a macrophage-specific CFTR knockout mouse. So one of the things they did was look at different alpha diversity indices for the intestinal microbiome. And so what I want to draw your attention to down here in these figures is that we look at the pan-CFTR knockout mouse. So this is the green here in these figures down below. We see that compared to the wild type, which is the yellow, we see that there is a significant decrease in all of these different alpha diversity indices, so including the number of taxa or OTUs in the samples. Looking at the Shannon index, which is a measure of species evenness within the intestinal samples, and also looking at the diversity of the samples using Faith's phylogenetic diversity index. The other thing I would like to draw your attention to in these figures is that when we look at the myeloid-specific CFTR knockout, so this is the purple in each of these figures, we see that when we compare it to the pan-CFTR knockout, so the green, for those indices, including the Shannon index as well as the Faith's phylogenetic diversity index, there's no significant difference between the pan and the myeloid CFTR knockout, which indicates that there is a contribution of the immune defect in CF to the development of intestinal dysbiosis. So what we wanted to find out was, what is the contribution of the loss of CFTR in the intestinal epithelium to the development of inflammation and dysbiosis? So we have these mice that have a floxed exon 10, and also crossed with these mice that have a Cre recombinase enzyme that is driven by the villain promoter. And these can be crossed to generate an intestinal-specific CFTR knockout mouse model. So these intestinal-specific CFTR knockout mice were kindly provided to us by Dr. Mitchell Drum and Dr. I don't know, I'm blanking on his name. Craig Hodges, my bad, from Case Western Reserve University. So thank you very much to them. And so we hypothesized that the intestinal-specific CFTR knockout mouse would develop intestinal inflammation and dysbiosis that is comparable to that of a global CFTR knockout mouse model. Okay, so in this study, we took intestinal-specific CFTR knockout mice and their sex-matched and age-matched litter mates. And so these mice were kept together until weaning, after which they were singly housed. And so these mice were put on either a diet that consisted of either the PEG osmotic laxative with a pelleted diet, or a daily provision of a nutritionally complete liquid diet, in this case Peptamen, and then regular water for a 14-day period. After this, we performed aseptic collection of fecal pellets, which we used to do our downstream analyses. So in order to look at intestinal inflammation, we measured fecal calprotectin, which is a product of neutrophils, and we measured this using an ELISA-based assay. In order to look at the composition of the intestinal microbiome, we isolated DNA from these stool samples and performed 16S ribosomal RNA sequencing with the help of the University of Missouri Metagenomics Center. We performed data analysis using PASS software as well as MetaboAnalyst. Now we look at the results of fecal calprotectin, so the concentration of fecal calprotectin in our wild type and intestinal specific knockout mice receiving both the PEG laxative as well as the liquid diet. We see that for both of these diets, our intestinal specific CFTR knockout mice have a significant increase in fecal calprotectin concentration in both of these diets, so indicating that increase in inflammation, regardless of diet, excuse me. Now we look at evaluations of the fecal microbiome. So specifically, we're looking at alpha diversity indices. So we looked at the Shannon H index, which looks at the evenness of the species in the sample, as well as Chow-1, which looks at the richness of species in the sample. And we see that for those mice that were receiving the PEG laxative, we did not actually see a significant difference between the wild type and the intestinal specific knockout in either the Shannon H or the Chow-1 alpha diversity indices. However, when we look at those mice that were receiving the liquid diet, we see that the intestinal specific knockouts have a significant decrease in both Shannon H as well as Chow-1 alpha diversity indices, so indicating an influence of diet. Now we look at measures of beta diversity, so comparing the composition of the microbiome between our groups. So for both the intestinal-specific CFTR knockout as well as the wild types. So we can see when we look at this visually using a principal coordinate analysis for both our mice receiving the PEG laxative as well as those mice on the liquid diet, the results of our permutational multivariate ANOVA reveal that there is a significant difference in the microbiome composition between the intestinal-specific knockout and the wild type in both of these dietary regimens. Now, when we drill a bit deeper down into what those differences are, so this is a stacked bar chart on the phyla level for those mice that were receiving the PEG laxative. And so here you can see on the left, this is the relative abundance of these phyla compared to one another. And we see the wild type on the left and the knockouts on the right. And I want to draw your attention to one of these phyla. So this is this orange here, the Actinobacteriota, which is increased in our intestinal-specific knockout mice. Excuse me. Now we go down to the family level for those mice that are receiving our PEG laxative. Again, with our knockouts here on the right. One of the drivers of this increase is this family member here, the Bifidobacteriaceae, which is this light blue here. And so this is increased in our intestinal-specific CFTR knockout mice. So even deeper down into this, this is a heat map that is showing us the top 50 most differentially expressed microbes in those mice that are receiving our PEG laxative. So again, we see knockouts here in the green on the right, and then the wild types in the red on the left. And we see that for several of these microbes, they have an enrichment of several microbes compared to the wild type. And whenever we look a little bit closer at this, we see, again, there's that enrichment of microbes in the genus Bifidobacterium, which are beneficial bacteria that aid in the digestion of fiber and also in the production of several healthy fatty acids. But we do also see this enrichment of this Clostridium inocuum, which is known as to be an opportunistic pathogen. Now, switching over to those mice that were receiving the liquid diet, this is again another stacked bar chart at the phyla level, looking at the knockouts on the right and the wild types on the left. I want to draw your attention to the phyla Firmicutes, which is, I believe, now called Bacillota. And so even though it's not significantly different between the wild type and the knockouts, when we look at the family level, we see that for the knockouts, there is this considerable enrichment of the family Clostridiaceae. And so looking at a heat map showing us the breakdown of the top 50 most differentially expressed microbes, again with the knockouts here on the right, wild types on the left. One of the things I want to draw your attention to with this heat map is notice that there is a significant decrease in the majority of these microbes for the knockouts as compared to our wild types over here. In addition, when we do take a closer look at those microbes that are enriched in our intestinal-specific knockouts, we see enrichment of several pathobionts, including several strains of Clostridium perfringens. We also see Clostridium difficile, and also, again, we see Clostridium inocuum. So again, overall, we do see this increase in several pathobionts. So in the mice that were receiving the PEG laxative, we did see that enrichment of Clostridium inocuum, as well as, even though I didn't point it out on the family stacked bar chart, we did see an enrichment of Peptostreptococcaceae, which is noted for its involvement in inflammatory bowel disease as well as colorectal cancer. So to go in a little bit more detail about Clostridium inocuum, so this has been noted as a potential diarrheal agent, specifically for those patients in which Clostridium difficile cannot be isolated. As well, it's been known to translocate into the tissue for those patients suffering from Crohn's disease. Now, when we look at the liquid diet, we saw that several species of Clostridium were enriched in the intestinal-specific CFTR knockout. So again, we saw Clostridium difficile was increased, and again, that's that diarrheal disease agent. We also saw Clostridium perfringens, which is noted for its involvement in causing food poisoning because of the elaboration of several different toxins. And again, we also saw Clostridium inoculum, which is that opportunistic pathogen that can cause diarrhea as well as exacerbations in Crohn's disease. So in conclusion, the results of our studies demonstrated that loss of CFTR function in the intestinal epithelium is sufficient in order to induce significant inflammation as well as dysbiosis in the intestine, and that these intestinal-specific CFTR knockout mice do demonstrate a less severe dysbiosis whenever they receive the PEG laxative as compared to the liquid dietary regimen. And that overall loss of CFTR in the intestinal epithelium is a major contributor to intestinal dysbiosis in CF. Okay. And so I'd like to take this opportunity to thank several people. So Dr. Sarah Young was the one who was the driving force for initiating these studies, as well as Estella Williams. She's a veterinary student that helped, collect a lot of the fecal samples, so she had the crappy job. Yeah. And then also Dr. Erickson, who has helped Dr. Clark and myself learn a lot about how to analyze gut microbiome data. And also, again, Dr. Craig Hodges and Dr. Mitch Drum. Sorry, I forgot your names, guys. Thank you again for providing us with the mouse model. Okay. And I'd be happy to take questions. So, yeah.
What she said. So please come up to the mic if you have a question.
Right.
So that was really a great talk. Very interesting. Are these mice grown in specific pathogen free? I mean, these pathogens are just coming into the colony.
Oh, yeah, so these are not specific pathogen-free mice, yes. So they came to us and they've been in our colony, and so they kind of came with what they had.
Okay.
Very nice talk. Question one, Alessio state. So it's, what is the difference between your PEG and the liquid diet? Because some of the difference may be introduced by the different components.
Okay, well, I'm glad you asked. Great question. So, yes, there is a considerable difference between the two diets as far as their overall composition. So in the diet in which they were receiving the PEG laxative, obviously there's the osmolytes and also the PEG compound that provide that laxative effect. But in addition, they receive a pelleted diet, which provides a variety of different soluble and insoluble fiber sources, which can go towards, supporting a more diverse microbiome. So that is a consideration to keep in mind, because as compared to the peptamen diet, it's a hydrolyzed whey protein diet. So its intent is that it's easily digested and absorbed for those individuals that have gastrointestinal diseases. So they just need that nutrition. So yeah, it's hydrolyzed. It also is fortified with lots of different vitamins and minerals in order to provide them with nutritional support. So those could have an effect on the different types of microbes that we see. So.
Yeah, very nice data. Two questions. One is, I don't know if you mentioned that, but both the wild type mice and knockout mice were kept on the same diet, meaning liquid diet or laxative.
Right.
And the second question is, what is the next step? I mean, are you trying to figure out a way to restore the diversity and the composition of the microbiota, like, for example, with, omega-3 or probiotics?
Okay, great question. Okay, so let me back up a bit. All right, so these, I think correct me if I'm wrong, the first question you asked me was like these mice that we use the litter mate pairs. So when they were weaned, they were all provided with PEG laxative and the pelleted diet. So for some of those studies, we kept them on the PEG, and for others, we transitioned them to the peptamen diet, right? Okay. Now, your question, which I can summarize as like, well, so what? Okay, they're different.
No.
I know. I'm sorry.
Because that's very interesting. So what are you?
That's okay. That's okay.
What is
All right.
your next step?
Next step?
No,
yeah,
I'm
right.
saying not what. I wouldn't never say that.
I'm sorry. I'm making you the bad guy. All right. Anyway, so that's a great question, right? So one of the things that interested me was that, with the advent of the modulator therapies and the correctors and whatnot, I was actually surprised to learn that these, at least as far as I know, for like long-term studies of people that have received these medications, it doesn't seem to have much of an effect on the intestinal dysbiosis and inflammation, right? So one of the things we wanted to know here was like, okay, so kind of seeing what the major contributors are to the development of the intestinal inflammation and dysbiosis. So it seemed that, we would want to see, for example, like if those mice that were receiving the peptamen diet, what if we gave them some bicarbonate in the peptamen in order to help with the solubilization of the mucus in their gastrointestinal tract? Would that help to kind of, fix that imbalance that we see, excuse me, in the different players in the gut microbiome? Like if we provided them with a soluble source of fiber or source of soluble fiber, so what can we do to kind of even the playing field, since loss of CFTR function radically changes the intestinal milieu? So what kind of interventions can you do in order to make things a little bit more, similar?
Thank you.
Yeah.
It's a good question for you. Ken Caldwell and others have shown that the fungome in the gut has a big effect on, bone marrow, granulopoiesis, and, even the composition of blood and then the ability of, for example, neutrophils or MACs to go into other areas. So, obviously fungi don't have 16S, but did you do 18S or ITS? And, is there any evidence for a change in the fungome in these mice?
We did not do that, but I would love to do that.
Awesome.
As well as like, I don't want to go too long, but, as well as like, we were also interested in looking at, the metabolome. So as well as like including analysis of the fungome and kind of looking at like a more full picture of like what exactly changes in these two conditions. And is there anything you can do to kind of, like I said, bring it back to normal, if you would. So.
Yeah, awesome. Well, one of the things that Caldwell did is that he sort of rewilded the mice and just introduced like a couple of fungal species.
Oh, yeah.
So that might be better than just having them roam around your your backyard.
Take your daily cup of fungus. Yeah. If you have any more questions for me, I'm also going to be at my poster for the first poster session. It's poster 349. Yeah.
Thank you so much. And Rowena's.
Okay.
She might also be performing comedy science later. So.
Okay, so next we'll introduce Carly Stewart, Dr. Stewart, to come present.
All right, thank you for having me and thank you for the introduction. Let's see. So I have no disclosures and I'll just hop right into it. So I don't need to tell you too much about cystic fibrosis, but I'll just bring it to the forefront of your mind. So cystic fibrosis, CF, is a genetic disorder caused by mutations in the CFTR gene. CFTR encodes an anion channel that transports both chloride and bicarbonate across epithelial membranes. And pulmonary complications are a major cause of morbidity and mortality in CF. Progressive loss of lung function is typically due to host defense defects. Some include impaired bacterial killing, abnormal mucociliary transport, and other defects. Today, I'll be focusing on abnormal mucociliary transport. So in understanding MCT and the defects in CF, we wanted to study MCT prior to the onset of infection and inflammation. And so since this is not doable in humans, we utilized a CF pig model to do so. And the CF pig model replicates the human CF lung disease. So I'm just going to give some background on some previously published data in the large airways of newborn CF pigs. So we were able to study the large airways of CF pigs usually using a CT scan imaging method where we use tantalum microdiscs that are about 325 microns in diameter. And we push these microdiscs into the airway with a puff of air, and they're deposited in the trachea and main bronchi. And then we can take a CT scan every 8 seconds to image the movement of these microdiscs over time. So here I'm just showing you an image of those microdiscs, a side view of the CT scan. So here, 8 seconds, 16 seconds, and 24 seconds. And the red arrow indicates movement with time. We can also see those microdiscs are embedded in mucus. So next, I'll just show you, since we're able to do this dynamically, we can watch this as a movie. So I'll show you a movie of a wild type newborn pig. The microdiscs are dark in color, and you'll be able to see these microdiscs with time clear from the airways. So here you see the microdiscs travel up the trachea and eventually clear out of the field of view. So we can also look at this in CF. And when we look at it in CF, we see something quite different. So I'm going to play this video, and what you'll see is things will start to move, but then be abruptly brought back down. So here, the microdiscs start to move, but then are abruptly brought back down. And this is thought to be due to defects in submucosal gland secretions. So submucosal gland secretions get stuck at the duct of the submucosal gland, and then as it gets pulled through the trachea, gets flung back down. All right, so what about the small airways and their role in CF? So we know that the airway tree is a fractal structure that increases in total cross-sectional area for efficient gas exchange, and that small airways make up most of the airway tree. These small airways aren't the easiest to study, but they may play a role in CF. So this is suggested by several observations in clinical studies. So old autopsy and explanted lung data have shown an increase in diameter of respiratory bronchioles, bronchiolitis obliterans, and destructive emphysema. Spirometry has shown that FEF 75 and 25 to 75 have been reduced and progress early in the disease, and that lung clearance index abnormalities in kids with CF are abnormal. Also imaging, the air trapping on CT scans have been shown, and then there are ventilation defects on hyperpolarized helium scans. So this led us to the question, is mucociliary clearance abnormal in the small airways in CF prior to airway disease? So previously, these observations have really just looked at obstruction. So we wanted to ask the question, is before that obstruction, mucociliary clearance abnormal? And so what we've seen in the large airways of CF is they're abnormal due to submucosal gland defects. So this makes us think, since the small airways have no submucosal glands, that it may be normal. So to be able to answer this question, we developed a dynamic PET scan. So I'll just briefly introduce what PET imaging is. PET imaging utilizes a positron-emitting isotope that can be delivered into the body and imaged from the outside. So the positron-emitting isotope emits a positron where it can collide with an electron. This collision makes an event happen where equal and opposite gamma rays exit and can be detected by a series of PET detectors. So what we did is we took gallium-68, which is a positron-emitting isotope with a half-life of around 68 minutes, attached it to microaggregated albumin, which is around 1 to 10 microns in size, delivered the radiotracer via a bronchoscope into the distal airways, and then acquired images before delivery and then continuously afterwards. So the good thing about dynamic PET is we're really capable of assessing at a really high temporal resolution. And then we analyzed the small airway with 2 approaches, which I'll talk about in a bit. So here I'll just show you a video of what one of those experiments looks like. So here is a wild type pig, newborn, it's spontaneously breathing. And so I'm showing an accumulation projection of the PET signal. So through the chest, I've accumulated all of the PET signal so I can show it on a 2D screen. So here you'll see signal go in and then things clear into the large airways. So first we looked at one region of interest, and typically nuclear imaging and mucillary transport looked at an outer shell. So that was our first step. We were able to create a 3D region of interest around the outer lung. And here's just a cross-sectional view of that. So the green is the region where we look at where the signal is leaving from. And we determined the thickness of this shell by using a micro CT of the porpoise lobe, and then looking at where small airways were less than 200 microns in diameter. So that has been previously defined in pigs as being a small airway. So when we look at clearance over time, what we found and was kind of excited about is that clearance occurs really fast. Here I'm showing you a cross-sectional image of the CT with the PET scan overlaid and the green outline of our region of interest. So at 0 minutes, you can see the PET signal within that region of interest, and then by 10 minutes, the signal is almost completely gone. And then if we look at that with time, we can look at the dynamics of the clearance. So here I'm showing you in blue non-CF pigs. So we had 7 non-CF pigs. On the x-axis, I'm showing time, and then the y-axis, the percent cleared from that region. So what we can see is within 5 minutes, a lot of the clearance occurs. And it kind of happens in 2 phases. So we see the first 5 minutes, there's this faster phase, and then the second, the last 10 minutes of the study, there's this slower, more plateaued phase. And if we look at the CF group, we see the phases still occur, but they're stunted. So CF pigs are able to clear less and have impaired small airway clearance. So a typical measurement for nuclear scans has been 20%. So we looked at what pigs cleared greater than 20%. Non-CF 4 pigs were able to clear more than 20%, whereas only 1 pig in the CF group was able to clear more than 20%. We also wanted to ask the question, does stimulating mucus secretion further impair small airway clearance in CF? So we know in large airways, stimulating mucus with methacholine decreases clearance. So we kind of wanted to see if this also occurs in the small airways. So we use a pernodrug agonist, UTP, to stimulate goblet cell secretions in the small airways. And what we found was that by applying UTP, we see a further impairment of small airway clearance. So here in purple, were CF pigs treated with UTP. So we also analyzed this data with an alternative approach. So our outer shell ROI includes not only the small airways, but also alveolar spaces. So it can be thought that material is getting retained within these alveolar spaces. So we decided to also do an alternative analysis to try to exclude these regions. So what we did was we took the airway and segmented out to the pleura. So we know that in the pig anatomy, it's a monopodal branching airway tree, which is different from a human, as in the main bronchus will taper down into a small airway. So we leveraged this anatomy in this next analysis approach. So I'll just slowly step through what we did here. So here I'm showing the distance from the pleura. So the pleura being at 0, and then as you go to the right, you're reaching the trachea. And on the y-axis, I'll show activity. So throughout the airway, we can see what the PET signal is along the airway tree. So here I'm showing you in blue the signal in the airway volume along this center line. And so we can see a lot of the signal is in the small airways. And so if we look at our airway segmentation and find where the diameter is less than 2 millimeters, so what CT can resolve, and take all of that signal and the area under the curve, we can look at another region of interest. So here I'm just showing at 0 minutes what that looks like. So the area under the curve from diameter of less than 2 millimeters. And then within 15 minutes, by the end of the study, this has decreased. And so we see a similar pattern to what we previously saw with our other region. But one thing that is different, in the non-CF, we see that there's a continued clearance. And we think this is because things were being retained in that other region. And then we also see a similar thing when we apply UTP. So in conclusion, we're able to look at MCT with high temporal and spatial resolution imaging from the dynamic CT, looking at more granule movements in the large airways, and the PET signal looking at the small regions, or small airway regions. We're beginning to understand MCT in the small airways, and we found that small airway clearance occurs fast and it's abnormal in CF, and that this may be due to abnormal mucus properties. Some future goals we have is to assess this with modulator therapy and or gene replacement therapy, and to develop this technique for use in humans. So both gallium and macroaggregated albumin have been used in humans before. So that's where we want to go. Thank you.
Okay, for questions, you can come to the microphone.
Hi. Is there any evidence at all of movement from the alveolar space to the airway?
Not that we could detect with this method, but that would be interesting.
Do you envision any opportunity to be able to evaluate the different airway clearance techniques that to date have rather crude volume of sputum production as the output? This might be a way to determine what, in fact, might be the most useful technique.
Yeah, that would be really interesting to use this as kind of an outcome measure. Yeah.
So what do you have to do now to get it to human?
Lots of paperwork.
But you're hopeful.
Yes, yes.
Hi, Emanuela Bruscia from Yale University. So I'm curious, what you see in the smaller way, could that be, how much it really depends also from the backflow that you have from the upper airways, right? Because you're showing very clearly that in your CF pig, you have a force that brings your microparticles down.
Yeah.
So you would think about it that in somehow this force is also propagating in the smaller way. Do you have a sense how this is impacting what is happening lower in the lung? And is your model able to try to
Yeah. So we have a really localized delivery. So we use a bronchoscope and then deliver the radio tracer very distally. But it may be that breathing might have an effect on transport, which is a future question that we'd like to answer. The mechanical effect of breathing may assist in small airway transport, but we have yet to be able to answer that. Thank you.
I have one quick question.
Sure.
This is in newborn pigs.
Yes.
Have you looked at other pigs?
We've looked at one older pig. Yeah. And a similar thing. Yeah.
Worst is just one. You don't know.
Yeah, just one.
Okay.
Yeah.
Next year.
Yeah. Yeah. All right. Thank you so much.
Okay, next we'd like to invite Dr. Paul McNally. Welcome.
Thanks very much. And thank you very much to the organisers for inviting me to give this talk on behalf of the Promise and Recover study groups. So, is this working, the clicker? No. That works, okay. Okay, so here are my disclosures. So just a little bit about the background to this data. So we know there's more and more talk about real world data recently in CF, and I think CF is a really good model for this. It started with ivacaftor when there was some knowledge that would be coming into clinical practice, and a few studies were set up, although the number then subsequently increased significantly for the dual combination studies, and there's loads and loads of real world studies for triple combination. So as a community, we've really copped on, and we're doing a lot of real world analysis, which I think is really, really powerful. And it teaches us a lot about the drugs themselves, but also about the disease. So just to give you an overview about Promise and Recover. Promise, as you know, is a big study in the US run by the CFF-TDN. It includes adult and pediatric patients. There's all genotype subgroups included. It's a very wide representation. There's lots of different sub-studies as well, and they're all reasonably sized too. And then Recover, a much smaller study in 8 centres in Ireland and the UK. It is adopted by the ECSF CTN. We just have F508 homozygotes and heterozygotes with the minimum function mutations, and these were based on the original licensing in Ireland and the UK, and we elected not to include other groups. And we're really focused on CT and LCI as important outcome measures in our study. We also have a subgroup looking at adherence, and some of this data will be presented tomorrow. We've less in the way of sub-studies. Okay, so in terms of methodology, so just bear with me, there's a little bit of data on this, but important to outline. I'll go through it reasonably quickly. So we combined the databases to increase, obviously, the power and generalizability. We sent anonymized data from Recover over to the Promise group, and Sonia Helchi and Morgan McCreary were in the CFF TDN Center in Seattle were responsible for all of the analysis. So we only sent samples that had adequate numbers of sweat chloride, so that the overall population size is a bit smaller. So we're going to use the nomenclature FF for homozygotes, FM for F and minimum function, FG for F and gating, and FO for F and other. So we included, because of the stability of sweat chloride measures in clinical trials, we included, we picked 6 months as the post-CTI measure, but we included everybody who had sweat chloride done between 1 and 6 months because of the stability of the measures. So we only reported age in years to ensure that we had a totally anonymous data set in the Recover study, so and a weight z-score. So we had to work backwards and do some backwards computing. So some of the results in terms of weight may not be 100% accurate because of this. But we don't think these are major errors. I'm not presenting much in the way of weight data here. I'm not going to read all this. This is just the methods we use to analyze the data, reasonably straightforward, and obviously all done by the CFF stats core. So we used OR, and we used the CDC OR package, CDC Anthro, for the back calculations of the Z-scores. Okay, so the results then. So this is just a kind of a look at the two different study groups, and I'll bring you through this again. Just the key here on the right to the acronyms and what they mean. So you can see that the RECOVER study has a much younger patient group, so a mean age of 14.8 compared to 21.5. And much more focused on younger subjects in terms of the breakdown. There was also an overrepresentation of the F508 homozygotes in the RECOVER cohort compared to the PROMISE study. And then if we look at baseline and 6-month change, you can see that the baseline sweat chloride, again, commensurate with what we've described above. The baseline sweat chloride was lower in the RECOVER study, and the FEV1 was higher. But then we saw similar changes in sweat chloride, FEV1, and CFQR between the two groups. So a reassuring, number one, difference in populations, which underline the importance of bringing them together. But number two, they weren't that dissimilar in terms of their response globally to ETI between the two groups. Okay, so when we look at the total sample size here, now again, we only exported data from RECOVER where we had a sweat chloride at baseline in 6 months, so we have 0% missing data, but obviously this is because this was pre-selected for participants who had data. The rate of missing data is about 10% if you look backwards at the numbers we excluded. And so we had a total of 755 participants and 707 who had valid sweat chloride measurement. Okay, so I just orientate you on the left-hand side. This is 6-month sweat chloride on the y-axis and age on the x-axis. You can see these plots as the data becomes less frequent. Certainly as we go to the right, you can see the error bars increasing. But if we look at the tighter part of the error bars, the RECOVER population is certainly younger, but the relationship between sweat chloride at 6 months and age is similar between the two groups, which is reassuring. Then on the right, we look at change in FEV1 and change in sweat chloride. And you might remember that the PROMISE group were the first to publish a correlation between sweat chloride change and FEV1 change. And again, we saw that in this group, but you can see that that relationship is fairly flat. And it's hard to know how clinically meaningful that is at a patient level. So then when we break down the baseline and 6-month sweat chloride on prior modulator use, you can see, and the red lines here are 30 and 30 down here and 60. And so you can see there's a couple of things that are of interest, I think, here. So without prior modulator use, obviously the baseline sweat chloride was higher, and a much lower proportion of those individuals ended up with a sweat chloride less than 30, with the bulk being between 30 and 60. And those previously on ivacaftor, then the majority of them ended up with a sweat chloride of less than 30. And you can see what looks like a bit of a floor effect in the middle there in the blue dots. And then on the right-hand side, those starting on a dual modulator, mostly obviously F508 homozygotes, a significant proportion of them ended up with a sweat chloride of less than 30 at 6 months, and very few actually with a sweat chloride above 60. Although I think it's a bit sobering to, if we look at all of the blue dots, there's still a lot of blue dots above 60 on ETI at 6 months. So we still have a considerable a population of people even on ETI who still have abnormal sweat chloride, and that's something that we can't forget about. Okay, so this figure just looks at the male-female differences in sweat chloride. So just to orientate you, on the left-hand figure, it's baseline sweat chloride on the y-axis, and then age group on the x-axis. So we broke this down into less than 12, adolescents at 12 to 18, 18 to 30, and then older adults aged over 30. And you can see reasonable spread of numbers across all of them. And then on the right-hand side, we looked at baseline sweat chloride by genotype group. And then again, red is female and blue is male. So what's interesting to note here is that there was no difference between sweat chloride among males and females at any of the different age groups at baseline or between any of the different genotype groups. So if you keep that in mind, when we look at the figures at 6 months, obviously you can see all of the sweat chlorides came down. But here we have on the y-axis 6 month sweat chloride and the age groups again, and you can see in the children less than 12, there was no difference between male and females. And then in all the other age groups, there was a significant difference with females having lower sweat chloride. It's particularly significant, obviously, in the 12 to 30 age group and just about reaching significance in the over 30 age group. And then when we looked at the genotype groups, it was most significant in the FF group, and we didn't see any significance in the F other group. But you should bear in mind that in the F other group, that's a very diverse, heterogeneous group of patients. So then when we look at, so we looked at baseline, we've looked at 6 months, so now we look at change in sweat chloride, and you can see in the children less than 12, there was no sex difference in terms of change in sweat chloride. And now in the over 30s, when we looked just at change, and there was no sex difference, but the sex difference was maintained in the people between the ages of 12 and 30. And then again, looking at change with the genotype groups, it was still significant for F508del homozygotes, but it wasn't significant for anything else. Okay, so just in terms of just looking at a graphical representation, and I'm not going to get into the statistical analysis of this because this was outside of the scope of the study. So in the upper panel here, you can see the various different age groups and they're color coded. And in the lower panel then, you can see male and female. So just a few things to note here. You can see that the sweat chloride at baseline is similar among all groups, but then it seems to be a little bit separated out in those less than 12. Who have a lower sweat chloride. An impressive FEV1 response in everyone, which again reflects what we saw in the clinical trials and perhaps is somewhat surprising in the broader scheme of things. In terms of CFQR, obviously there seems to be a little bit of a ceiling effect as you get close to 90 here with less improvement in the under 12 age group. And then you can see a clear separation between male and female in sweat chloride. At 6 months, not so much at baseline. And then interestingly, we haven't looked at this data, but we plan to. You can see that the baseline, so that the 6 month values for FEV1 and CFQR are reasonably similar between the groups, but the females came from a lower baseline. And so that's certainly telling in light of the sweat chloride changes. Okay, so when we then separate out the groups and look at the group who achieved a higher sweat chloride and the group that achieved a lower sweat chloride, and we look at these groups, you can see that about 25% of people overall achieved a lower sweat chloride. And among these, children less than 12 were overrepresented. Females were overrepresented, and people with the FF genotype were overrepresented, and the opposite is true of the minimum function heterozygotes. And then when we look at change in sweat chloride from baseline, obviously the group who achieved a lower sweat chloride started from a lower baseline, but not only that, they also had a better sweat chloride response with an average of less than 50 compared to less than 40. So it's not just the baseline. So then we used a logistic regression model to look at the odds ratio of achieving a sweat chloride of less than 30 6 months post-baseline. So starting from the top here, you can see that the F other and the F minimum function and genotypes, they had a lower odds ratio of achieving a sweat chloride of less than 30. And it was most significant really for the FM group, which are a much more discrete group. There was an overlap, obviously, between the F gating and the FF groups, as expected. And then a very strong odds ratio of over 2 for females. So females more than twice as likely to have a sweat chloride of less than 30 at 6 months. There was also for every change in decrease in sweat chloride of 10 millimoles, there was a 1.65 odds ratio of getting a sweat chloride less than 30. There was no relationship between baseline FEV and the odds ratio. And for every 10-year decrease in age, there was an odds ratio of 1.5 of achieving a lower sweat chloride. So the groups that are most likely to have a sweat chloride, a significant correction to less than 30 of sweat chloride are female, younger, homozygous for F508del, and with a lower starting sweat chloride concentration. So the million-dollar question is, what is the explanation for this? So the genotype effect, I suppose, is not that unanticipated, but the sex effect was a little bit of a surprise. And so it's hard to know what that is. We've discussed this a little bit, and adherence may be an issue because we didn't see it in the children less than 12. Clearly, the 12 to 30 age group, you have to think about hormonal influences, and that might be the case. And it may also be to do with something like pharmacokinetics. So we know what the data says, but we don't know why yet. So there's a couple of important remaining questions. So, the differences in outcomes in females. Like, do these persist, and do they relate to do the difference in outcomes? Are there differences in outcomes, and do they relate to the sweat chloride, the initial change in sweat chloride? We know from cross-sectional studies that sweat chloride levels are different in people at different ages, and they tend to be higher in older people and lower in younger people. But we really don't know what happens when you start a modulator. What happens to your sweat chloride, and does that change? So we'll be able to find that out from studies like PROMISE and RECOVER. And the question is, do the lower sweat chloride levels translate into future outcomes, and how is this, and how do we work this out? We're going to try and see, certainly from the RECOVER study, where we're looking very closely at adherence, we're going to try and see if we can establish if adherence is involved in changes in sweat chloride response. And certainly, this example, I think, of a really great collaboration between the two studies, where we're able to get significant power from such a large patient cohort, and there's much more we can do with this in the future. So just want to thank the study teams. There's huge study teams in PROMISE and RECOVER. Particularly on our side, I just want to thank Karen Lester and Rachel Cregan. In particular, the participants and their families. It's a huge endeavour, actually, to stay involved in studies like this, particularly as they go on, and we're really so grateful. And finally, to the CF Foundation, who provided the funding for both of these studies. Thank you.
Thank you so much, Dr. McNally, for that excellent presentation. It's 10:28, so I'm not sure if you had time for a question.
Yeah.
Okay. All right. So for Dr. McNally's speaking elsewhere, so you have to rush to the mic to ask your question before he's running.
Hi. Clark, Missouri. Very nice talk. Is there any data on the relative skin thickness having a role in terms of the perhaps being cooler at the sweat gland that allows a more permissive environment for delta V508 to come to the membrane?
I have no idea. I'm not an expert in skin or the performance of sweat chloride. I just don't know the answer to the question.
Yeah, lower temperatures are more conducive for CFTR delta V508 to make it to the membrane.
All right. Okay.
Hi, Isabel Sami Paris. It's more a comment on the very, very interesting sex effect on the sweat chloride change in the post-pubertal females, because I was wondering, because it has there is a literature about the estrogenous correcting effect on CFTR, delta 508 CFTR. So maybe this could be one of the explanation. And I think it would be interesting to have longitudinal evaluation of the sweat chloride according to the female cycle, and to check if we have, according to the estrogenous blood level, change in the chloride, in the sweat chloride.
It's a really interesting question. Yeah, I think it's probably beyond the studies as currently designed, but it's a good suggestion. I think we want to try and get to the bottom of this somehow, and perhaps that's a suggestion for a future study.
Can you just comment on if you're in the recover cohort, if there's other gendered factors that are being collected beyond adherence? So like health behaviors, health access, financial status, like just, you would think, I get that in most CF studies we collect sex data, but it's perhaps the gendered factors that are also playing a role beyond adherence.
No, we're not. And I'm not sure, Sonia, do you know in PROMISE if that data is being collected?
No, I was thinking, in PROMISE we know when the menstrual cycle is for women in PROMISE. So that may be something to think about. While not longitudinal, there is a point later in the cycle.
Any other questions?
There are also morphological changes to the sweat gland with exercise. Is there exercise data for these patients?
No, we don't have exercise data in recover.
You might end up seeing more sweating from people who do more exercise. I don't know if that makes fluoride level.
Yeah. Me neither.
Sounds like recover 2 and promise 2.
Yeah.
Thank you so much.
Thank you.
So next we'll invite Dr. Susan Burkett. Welcome.
All right. Thank you very much, everybody. Thank you to the organizers for letting me give this data today. My lab has been working on the effects of ivacaftor, specifically ivacaftor on infection, using the rat model for a few years now, and so I'm happy to share those data today. All right, so I have no relationships to disclose. All right, so some of the first data that came out of the original goal study was really focused on the lung burden of pathogens common to CF. And one of the first things we found was that the Pseudomonas incidence decreases in patients with ivacaftor therapy. This has been followed up much more recently by a paper led by Pradeep Singh and the PROMISE group, who all found that the lung burden of Pseudomonas and other bacteria, or rather the sputum density, drops as patients are on triple combination over a period of about 6 months. But we can see in the graph down below that the bacteria are being reduced by modulators, but they're not being eradicated from the lung. And so we really wanted to understand why that was. So my lab has been using CF models of rats for some years now, specifically because of this phenotype we see in the graph on the left with mucociliary transport. This is really a progressive feature of disease in the rat. You can see that at 1 month of age, mucociliary transport is normal, but by 6 months of age, it's severely delayed. Now, what's interesting is that when you infect rats across all ages with Pseudomonas, even using a clinical mucoid isolate, they really only stay infected at 6 months of age when they have mucus in their lungs before infection. So to understand the relationship between CFTR and mucus and infection, we use this 28-day model where we infect with mucoid isolate of Pseudomonas. We do use agarose beads, and then we take time points at days 0, 3, 7, 14, and 28. Now, for a rodent model, the acute phase of infection really occurs in like days 0 to 7. This is when the animals are most obviously sick. By days 14 to 28, we're really looking at more of a chronic phase of infection. So when the rats at 6 months are infected, we can pull the lobes from their lungs and count CFUs from the lung tissue itself. We see in the wild type and the open circles that there's plenty of bacteria at day 3. The dotted line is the level of inoculum. But by day 7, the wild type rats have completely cleared the bacteria, even with the agarose beads. The CF rats have a different story. They again have plenty of bacteria at day 3. With a drastic reduction in the lung burden by day 7, but that slowly climbs back to the level of inoculum by day 28. So there's a couple of things to note about this infection model that may help put some of the rest of the data in context. The rats are clinically sick, like visibly sick, between the 3 and 7 day time points. This is when you see them hunched and ruffled and dyspneic. But by day 14, they really have recovered any clinical signs. So by day 28, what we think we have is much more of a chronic, stable infection with Pseudomonas. Now, we've done a lot of outcomes from these infection models, and that's all published if you're interested, but we've consistently used the presence of mucus as a marker for infection. You can see that in the 6 months at day 28, we have significantly higher MUC5B and MUC5AC in the CF rats. And by day 28, the rats have developed these mucoid mucus plugs in their small airways. This is very consistent throughout the lung. So in order to understand how modulators were affecting this time course, we created a humanized G551D model. This uses the human cDNA that's been inserted into exon 2 of the rat endogenous gene. So it uses the rat endogenous promoter to express, just as it would the rat sequence, with the G551D mutation. This ensures responsiveness to ivacaftor. And the gene expression of both the rat sequence and the human sequence was tested in these models. We can see an abundance of human sequence, but very little rat sequence. And when I found some cross-reactive primers to look at mRNA expression, we found that the rats are making about as much CFTR mRNA as the wild type. And then just to confirm that there were no PK issues with ivacaftor, we tested the pharmacokinetics of ivacaftor in this model. So like many of the human studies, we see that the drug does tend to partition to the tissue. This is the rat lung lysate, and as well as the epithelial lining fluid. So importantly, although it looks very small here, even the plasma concentrations are well above the EC50 that's been reported. So when we look at CFTR correction in using ivacaftor in this model, we took trachea from G551D rats that had not yet been exposed to ivacaftor. That's in the blue line. You can see that as we start to add VX770, the current increases in a dose-dependent way until we add the CFTR inhibitor 172. Now, what's interesting about the wild type is that the activity, the change in activity after forskolin is very dramatically high, and we don't really see an increase with VX770. This is likely because all of the CFTR has been opened by the forskolin, and there's not much left to potentiate. Now, when we add CFTR inhibitor 172, again, we see a very strong signal. And when we look at the summary data, we get about 50% of correction in the G551D compared back to the wild type. And we can also correct the mucociliary transport. So these are representative images of OCT imaging of exogenous trachea. The red bar marks the airway surface liquid depth, and then you can see the highly dense regions of mucus above, marked by the MU. Now, when we quantify this, we can see decrements in airway surface liquid depth, periciliary layer depth, mucociliary transport that are all corrected by the addition of VX770 or ivacaftor. And then when we looked at the effective viscosity, also in situ, we saw that it was very high in the G551D rats, but restored to normal with ivacaftor administration. All right, so we know that the ivacaftor corrects the CFTR and the mucus, and we hypothesize that in the absence of any other complicated factors, because these are, again, kept in a very clean facility, unlike people who are walking around in the world with lots of diseases, we thought that we could ameliorate infection using ivacaftor. So up in the left, I have the kinetics of the rat infection, just to remind you what that looks like. We did 2 infection schemes using the treatment approach. Essentially, we infected the rats at day 0, and then we did a short ivacaftor treatment during the acute phase that ended on day 7. And then we did a longer treatment that ended on day 28 for the chronic infection. And what we found here was a small decrease in CFUs in the upper left. The green boxes are the rats treated with ivacaftor. At day 7, there's a small decrease in CFUs, but that goes back to match the vehicle-treated rats by day 28. We didn't see any difference in the number of neutrophils or the percentage of neutrophils infiltrating into the lung, nor did we see any difference in MUC5B at either time point. We did see a decrease in MUC5AC at the acute time point, but again, that's back to match the vehicle by day 28. So we decided to try a prevention approach. So many of the patients that have started modulators are already infected, but the younger children who are coming into age might see the modulators before they see infection. So we decided to ask if this would prevent infection in this model. So in this case, we did 3 different time points. We started ivacaftor dosing 7 days before infection, and then took time points essentially at day 3 post-infection, day 7 post-infection, and day 28. This time we did see a more dramatic difference in CFUs in the green boxes. We can see a decrease at days 3 and 7, but again, by day 28, this is back to the vehicle. This time we saw a decrease in the neutrophil influx at days 3 and 7, but again is back to the vehicle by day 28. And this time we actually saw more MUC5B and 5AC by the chronic time point in rats treated with ivacaftor. So to see if this affected the mucociliary clearance, we looked at G551D rats either with vehicle or ivacaftor during both the acute and chronic phase. These are the representative images. And when we measured airway surface liquid depth, we actually saw that G551D rats who are infected and treated with ivacaftor, this is in the far right in the green triangles, they had ASL depths that was no different than the wild type. But there was a difference in mucociliary transport in this group. Again, the mucociliary transport is defected compared to the wild type, but it's no different from the G551D, either untreated and uninfected or untreated and infected. During the chronic time point, we've actually lost our ASL benefit in the G551D treated group. You can see that in the representative images down in the bottom right. And in this case, we did have a slight restoration of the mucociliary transport. In the infected group, it's higher in the ivacaftor treated rats compared to the vehicle treated, and is actually not statistically different than the wild type. So what about the CFTR current? So we did want to check and make sure that ivacaftor was actively correcting CFTR in this infection scheme. So to test, first of all, what happens to CFTR correction during infection, we looked at wild type, uninfected, at day 3 post-infection and day 28 post-infection. So if you remember, the day 28 wild type rats had completely cleared infection and their lungs should be back to normal. So when we look at the forskolin dependent change, we see a big increase in current after forskolin in the uninfected rats, but we actually see a very small change in the rats infected at day 3. At day 28, the forskolin change is back to the uninfected. Now, when we look at the CFTR dependent change, which indicates the amount of current that is specifically due to CFTR, we actually see no difference between the groups, no statistical difference. So this suggests to us at the acute phase in the wild type rat that all the CFTR is open and not amenable to further activation with forskolin. So we moved into our infection model with the ivacaftor treated rats. So here we're looking at wild type treated with both with vehicle and ivacaftor, and G551D treated both with vehicle and ivacaftor, all at the 28-day time point. So here we see the same forceful-independent increase in the wild type. We actually don't see that with the ivacaftor. Again, we suspect that the channels are all actually open and unavailable for pharmacologic agonism. In the G551D, we see no forceful-independent change in the untreated, which is what we expected, and we see a high forskolin response in the ivacaftor-treated rats. When we look at CFTR-dependent change, this time we still see a high difference in the wild type, which is actually slightly reduced with the addition of ivacaftor. But this time with ivacaftor treatment during infection, we actually see a change in CFTR current that equals that of wild type. So you remember before infection, there was only about 50% of wild type. So we do see a higher CFTR activity during infection. All right, so what does all this mean? So basically, we didn't really see what we expected or what we hoped, which is that ivacaftor didn't really prevent infection, certainly not in the chronic phase. There are some changes to the infection characteristics during the acute phase, which actually may suggest that they are less amenable to the types of exacerbations that patients get. And we've seen that in the patient trials as well. At the chronic time point, unfortunately, any benefit from ivacaftor appears to be gone, both in the prevention and in the treatment experimental schemes. However, correction of CFTR activity through modulators is preserved throughout the infection. The modulator correction of the mucociliary clearance defect is mitigated. It's a little bit different than in the uninfected rats. So overall, it does seem like infection, at least with Pseudomonas, appears to be decoupled from mucociliary clearance and CFTR function, especially during chronic infection. All right, so moving forward, we really want to understand why this is. So we're going to dig into the inflammatory response. We've started looking at cytokine profiles to see if there's anything different. So far, we haven't seen much. We are going to look as well with combination therapy, adding mucolytics or potentially antibiotics to see if that changes anything. We know a lot of those data from the patient experience as well. But we are interested to know if this is specific to Pseudomonas or if this is common to other pathogens. So we're working on a Staph aureus model as well. All right, and I just want to acknowledge my lab. This group in the red is the current lab that does a lot of work, a lot of rats, a lot of bacteria. And then in the gray, I have former members who have helped with this project a lot. The CF Research Center at UAB has been very supportive under a number of directors now. And then Megan Kudrowski has helped a lot with the infectious outcomes as well. And then I've received funding from the foundation and the NIH. With that, I will take any questions.
I was wondering if you had plans to look at histology to see if you have more mucus cells and less capacity for mucociliary clearance because they're fusillated cells, or looking at bronchiectasis. What has the infection done to the structure of the lung that might explain some of the reason that ivacaftor isn't correcting the mucociliary clearance by 28 days?
Yeah, so we have the histology done. I think, if I remember correctly, we see some clearance of the mucus plugs, but we have not yet looked at the distribution of cell types, which is sort of the next thing I'm interested in. We can see, at least on histology, how many cells are making mucus using ABPAS, and I think that could be one of the things that's different about the animals, yes.
David Stoltz, University of Iowa. Real nice work, Susan. So correct me if I'm wrong, but I think you're measuring MCT sort of in the trachea, right. The electrophysiology. Most of the infection is probably distally.
Right.
So two questions. One, do you think there's sort of it sort of follows up a little on that question, sort of a communication? And what does the trachea look like histologically in this chronic infection model?
Yeah, so the trachea, by 28 days, often looks fairly normal. We do see a lot of mucus accumulation at previous time points in the infection. As far as how many cells are making mucus and what that cell type distribution is, that we haven't done yet, and I'm interested to know if that's different. In the ivacaptor treated rats. Now, in the uninfected rats, what we see is the trachea really does mimic the smaller airways where we have more mucus, even if it's not as occlusive in the later time points. We have more mucus producing cells, and it does seem to match a lot of the smaller airway pathology that we just can't measure using OCT or using chamber.
Hey, Susan, UNC, David Hill from UNC. Great work. Did you look at DNA accumulation at the 28-day time point that would correlate well through neutrophil data?
Yeah, that would. So I think we've run that once or twice, and we didn't find anything striking. One of the challenges with the neutrophil data, I think, is that the infection does get localized enough that there are airways that are clear and airways that are packed with mucus and neutrophils. And when you do the bronchoalveolar lavage, of course, that all gets lumped in together. So we didn't find anything striking with the DNA, but we also haven't looked specifically at areas that are impacted.
Hi, Susan, Wanda O'Neill. Do you see any evidence of bronchiectasis in your chronic model?
So we have seen bronchiectasis, but we have to push the animals much farther past 28 days. We often have we need either multiple infections, multiple pathogens, or just a longer time point. One of my students has a poster. I don't know what number it is. I should. I'm a bad mentor. But we do have a poster showing some bronchiectasis, bronchiectatic changes, but it really does take a good bit more length of time than 28 days to achieve that.
Susan. Here.
Hi.
Where are the bugs hiding? Do you know?
That is what I'm trying to figure out. So I know from other experiments, not ivacaptor experiments, I know that we can clear away the mucus plugs and not change the lung burden, which means they have to be someplace besides the mucus plugs. Whether that's on the epithelial surface or whether that's some other place that we just haven't found yet, we haven't figured that out. I'm interested to know if they're either at the epithelial surface or if they're moving into some cell type like a macrophage and just not being cleared, and to keep that infection around. Because we really thought that we could clear the mucus plugs and clear the infection, and that just didn't happen. So I really want to know that, yeah.
Any more questions? You're off the hook.
Thank you.
So because we're running quick and we've chosen speakers that need to run between rooms, I think we'll just wait on Dr. McIlvaney for our last talk. And so in the meantime, you have the privilege of standing up and doing a stretch if you'd like. Yeah. Rowena, you want to come do stand up? Do you need this water? You were coughing. There you go. I tried to get your attention and it was like, I hear it's just so dry, right? Of course.
Now it's written everywhere on the machine of the Jason, all the people have typed it about last
Great job. Oh, that got out? All right. Yeah.
I don't know exactly.
That's right.
But I don't think it was
Yeah, I don't either. No.
Chris does, but who knows?
Chris is right there, actually.
Chris?
Well, you're the unfortunate thing, so what happened?
Well, now that I have it for
Right, and so we got caught in So I guess going first, you get like open, you know, people have like said they.
I don't freeze, I don't your stuff. That's not. Yeah.
You just get it ready wrapped up.
Next time, I had like house.
No,
So that's why I wanted to do that. Yeah. Probably was.
Yeah.
Now we have
Yeah. Okay.
Nice. No.
All right, the man of the hour has arrived, so please take your seats. Welcome.
oh,
Hi, everyone. I'm terribly sorry. I was chairing a session upstairs. I hope I wasn't keeping everybody waiting unduly. Okay, so we might get started. Now, so we know that pulmonary exacerbations of cystic fibrosis are important. They drive morbidity, loss of lung function, reduced quality of life, hospitalization, death, and healthcare costs in people with CF. But despite this, these inflammatory events have been relatively understudied compared to the more chronic course of CF lung disease. And perhaps by extension, there's a lack of effective anti-inflammatory therapies for exacerbations, and that represents a treatment gap. Corticosteroids are potent, widely available, and affordable anti-inflammatory drugs. They inhibit cytokines, chemokines, adhesion molecules, as well as enzymes and receptors as well, obviously. But the randomized control trial data for steroids and CF are limited and largely confined to non-exacerbating children. The studies that are available tend to lack consistency in terms of dosing, treatment duration, and adverse event reporting. It's also possible that some cautionary tales from pediatric CF have deterred investigators from trialing steroids in adults. Perhaps the best known example of this is the 1995 study by Eigen and colleagues that randomized 285 patients to placebo or alternate day oral prednisone. These investigators found that prednisone ameliorated lung function decline in CF children colonized with Pseudomonas compared to placebo, But these results were tempered somewhat by undesirable effects on glucose metabolism and impaired linear growth. And some of you may recall that the high dose arm of that study, the 2 milligrams per kilogram arm, was stopped early for harm. So we wondered how we might help figure out the steroid question for exacerbating adults. And we decided to utilize STOP2 and perform a secondary analysis of it. STOP2 is a large, pragmatic, randomized trial of antimicrobial treatment durations in adult CF exacerbations. And that was to investigate a potential role for steroid in the management of these events. Briefly speaking, that the outline is in broad terms on the screen here. STOP2 involved 3 study visits, the first of which coincided with the start of IV antibiotic therapy. The data collected at each visit included spirometry with lung volumes, routine serum biochemistry, and respiratory symptoms as assessed by the CRIS. 982 participants were randomized to an antimicrobial treatment duration at visit 2, and that occurred about 7 to 10 days after the initial commencement of IV antibiotics. For the purpose of randomization, participants were categorized as either E or ORs, as you see on the screen, early robust responders, and they received either 10 or 14 days of IV antibiotics, or non-early robust responders, NE or ORs, who were randomized to either 14 or 21 days of IV antibiotics. And for the purpose of this study, an early robust responder was defined as a patient who had an FEV1 increase of at least 8 percentage points, as well as an improvement in symptoms of at least 11 points on the CRIS at visit 2 compared to visit 1. And then visit 3 occurred 2 weeks after the scheduled completion of IV antibiotic treatment. It's worth noting that candidates were excluded if they'd recently been treated with IV antibiotics or if they were receiving chronic corticosteroids. And importantly, they were also excluded if they had a suspected diagnosis of ABPA, the treatment of which, of course, frequently involves corticosteroid therapy. In terms of outcome measures, the primary endpoint and the secondary endpoints were aligned very closely with the original parent study, the STOP2 study for antimicrobial treatment durations. The primary endpoint was the absolute change in percentage predicted FEV1, and our secondary and safety endpoints included symptoms as assessed by CRIS, weight, FEV1 in litres, C-reactive protein, adverse event frequency, and time to next exacerbation, specifically median time to next pulmonary exacerbation requiring IV antibiotics. Now, it's worth bearing in mind that corticosteroid use in this study was at the discretion of the treating physician, and therefore unadjusted comparisons between those who received corticosteroids and those who did not were vulnerable to bias. And to try to address this, we used a propensity score matching approach. We used multivariate logistic regression to combine pre-specified adjustment covariates and clinical characteristics hypothesized to predict treatment with steroids to generate individual propensity scores. And this approach also provided the opportunity to match for factors such as randomization arm when assessing the treatment effect of corticosteroids on outcomes. 2 distinct propensity score matching strategies were employed in this study. The first was a permissive or greedy matching approach whereby every steroid-treated participant was matched to the nearest untreated participant, irrespective of the magnitude of difference between their propensity scores. So it didn't matter if they were a good deal apart, the nearest person they were matched to. And treated participants in this approach could also be compared to multiple controls in the event of ties. So in other words, we were trying to cast the net as wide as possible to include as many reasonable matches as we could. The second strategy, on the other hand, restricted matching to a single comparator participant with a propensity score difference of less than 0.03 to eliminate unsuitable matches. This is one where we kept it tight in order to preserve really high quality matching. Of the 982 pulmonary exacerbation events captured, 168 of these individuals were prescribed corticosteroids, 122 of whom got them before randomization, along with their initial antibiotics, and 46 who got them after randomization. Overall, patients who received corticosteroids were more commonly female and were more likely to have established obstructive lung disease. They were also older on average. The groups were otherwise reasonably similar. Nearly half the trial participants prescribed steroids were homozygous for F508del, of whom 59 received corticosteroids before randomization. And more than half were positive for Pseudomonas in sputum or bronchoalveolar lavage fluid at the commencement of IV antibiotic therapy. And again, no ABPA in the cohort. Now, in terms of prescribing practices, this varied quite markedly between centres. Here we see the percentage of patients per site who got pre-randomization corticosteroids, excluding sites that didn't prescribe any at all. And we observe a range here, anything from less than 5% at a given site up to 75%. And here we see the contribution of each steroid prescribing site to the total trial population. So this ranged quite a bit as well. The bottom line here is that we didn't have a situation whereby a single center contributed half the steroid-treated population, for example. So then we went on and looked at the potential factors influencing the decision to prescribe corticosteroids using a multivariate logistic regression, and found that initiation of corticosteroid treatment prior to randomization was associated with a few things. In particular, an age of at least 30 years, female sex, baseline FEV1 less than 50% of the predicted value, and prior ABPA, even in the absence of an active ABPA exacerbation. We were interested to find there was no relationship whatsoever with CRP. It wasn't predicted by Pseudomonas or frequent exacerbations either, and we thought that was interesting because corticosteroids are essentially an anti-inflammatory therapy. When assessing post-randomization steroid prescription, we excluded variables such as exacerbation frequency, modulator status, visit 1 CRP, and ABPA history. And we did that based on the premise that their influence on physician behavior would be largely confined to the initial phase of exacerbation management. And in their place, we added the presence of a persistently elevated CRP at randomization and this NE or status, as well as adding in high pre-randomization prescribing percentage at the site the patient was at. And for this group, the only association was with NE or status, and that suggested to us that corticosteroids were being used as a salvage therapy after a poor initial response to treatment. When it came to the primary outcome of change in percentage predicted FEV1, adjunctive corticosteroid therapy was not associated with greater improvement compared to standard treatment without steroids. Panel A here on the left shows the comparison when strict propensity score matching is applied, and this is for pre-randomization steroids. The same effect is observed when it comes to steroids given post-randomization, whereas panel B on the right shows the treatment differences in response to steroid when we use the more permissive matching approach. For those not familiar with the ATE and ATET in panel B, they stand for average treatment effect. That's essentially the average marginal treatment effect in the total population, and ATET is the average treatment effect on the treated, and that's the marginal treatment effect in the subpopulation that received the treatment. So again, no difference. And whether you were an early robust responder or a non-early robust responder, the outcome really didn't change. This is the sensitivity analysis for the association of pre-randomization steroid with changes in percentage predicted FEV1, whether you were an EOR or an NE no difference. Same goes for symptoms. When we used strict matching, we saw no difference in symptomatic improvement, irrespective of when steroids were given. For the more permissive matching approach, we did see a small but significant average treatment effect difference in terms of the CRIS and post-randomization steroid patients, but this wasn't a clinically meaningful difference. The MCID, the minimal clinically important difference in CRIS, is an improvement of at least 11 points. So when we asked the question of whether steroids were associated with a CRIS change of 11 or better, no difference. Same thing for CRP also, which might be a surprise for some, but bear in mind that the CRP endpoint in this study applied to visit 3, which was 2 weeks after the cessation of IV antibiotics. And similarly, no change in weight, regardless of whether we kept it tight with the strict matching approach or whether we cast a wider net with the permissive approach. Adverse events were not increased in corticosteroid treated people with CF. The total number of serious adverse events, however, was higher among patients receiving corticosteroids. We're comforted to know that the number of corticosteroid-related or pulmonary exacerbation-related SAEs, for that matter, were no different between the groups. When it comes to time to next exacerbation, we ascertained the data from the US CF Foundation patient registry, as well as review of site-level records, and followed these individuals out as far as 1,200 days. And panel A on the left here, we see people with CF who commenced corticosteroids pre-randomization compared to all people with CF who did not receive corticosteroids at any stage. So this excludes the post-randomization individuals. And there's actually a significant difference between the groups. The hazard ratio is about 1.3, which initially suggests a shorter time to next exacerbation in steroid-treated individuals. But when we apply propensity score matching, as we see in panel B, that effect disappears. So panel B is the more reliable result in this instance. And these are the within-year data for time to next exacerbation. Same exact pattern, and no significant difference when we match. So what can we say about steroids in the STOP2 cohort? Well, in patients receiving IV antibiotics for acute CF pulmonary exacerbations, early co-administration of systemic corticosteroids did not result in improved clinical or biochemical outcomes. And we saw the same thing for patients who received corticosteroids more than 1 week into their treatment course. There are several potential explanations for this. The most obvious, I suppose, is that the systemic corticosteroids given here were insufficient to overcome the inflammatory burden in some individuals, but there's also the possibility, particularly given the treatment was not guided by inflammatory status, that there was just simply an inability to identify good candidates for steroid therapy on the part of some of the treating clinicians. And that highlights, again, the importance of doing randomized control trials in this area that are prospective and steroid focused. In terms of the limitations that are associated with the data and what might give us some caution when interpreting the results, the biggest thing, I suppose, is that propensity score matching carries with it the risk of omitted variable bias. And further to that, inferences based on propensity score matching data are limited to subgroups in which balance is achieved. Now, that latter point doesn't really apply here because we have good balance in the groups, but the first point does hold. There was also incomplete CFTR modulator coverage within the group. Overall, however, I think when it comes to these two limitations in particular, I think it's fair to say that these limitations would, if anything, be expected to promote a positive treatment effect. So I think we can stand by our conclusions for the most part. In addition, pre-specified adverse events of special interest were limited to those pertaining to commonly used antibiotics, because this, after all, was a parent study that was focused on antimicrobial treatment durations. So again, if we were to look at this prospectively, we'd want to know a little bit more about some of the rarer but steroid-specific adverse events. I will say that when it came to things like hyperglycemia and so forth, while that would be expected in individuals receiving steroid and indeed individuals with CF diabetes, we were again reassured that no clinically significant changes in blood glucose status, which would have been captured by the original study, were recorded. And then finally, we also used an event-based definition of pulmonary exacerbations, which ran the risk of missing some of the more subtle or softer events, but also enabled us to focus on events that are more closely associated with mortality, morbidity, and healthcare costs. Just before I wrap up, I'd like to pay a special word of thanks to the STOP2 steroid investigators, in particular the folks on the screen here. Chris Goss and Patrick Flume are the co-PIs of the study, based at the University of Washington, where I'm at, and the Medical University of South Carolina, respectively. Sonia Helshi is at Seattle Children's and the University of Washington, D.B. Sanders at Indiana, Natalie West at Hopkins, Barbara Fogarty at the CF Foundation's Therapeutic Development Network in downtown Seattle, Dutch Van de Vander over at Case, and Katie Odom Davis at Seattle Children's as well. So thanks very much, and I'll take any questions you have.
Thank you very much. Please come up to the stage or the microphone for the questions. Or the stage. The stage works.
Hi, Peter Walker from Melbourne. There's a bit of evidence now about predicting response in COPD using blood eosinophils. Do you have any data on baseline and subsequent blood eosinophil counts?
Yeah, I think it's a big question for this study. The answer is that it's incomplete data. So we do have access to some site level records, particularly at the University of Washington and some of the other sites that were heavy recruiters to this study. What I would say is that if it were the case that eosinophil counts were higher in individuals who were prescribed steroid, or indeed if these individuals had, say, concomitant asthma or other overlaps that might suggest a steroid response, if anything, we'd expect there to have been a treatment difference between the groups rather than a lack of a difference. But I still think that your point is well made. I still think that ultimately pre-selecting, say, or including, say, as a stratification criteria in a future study, things like eosinophil counts when it comes to steroids would be very important.
So while we're waiting for other questions, I'll ask a question. I'm curious in terms of, you listed the factors of what were associated with pre-randomization steroids, and there was a number of sites that used steroids at that stage. And do you think it warrants more investigation, like whether it's through a survey or qualitative interviews with some of the prescribing physicians as to whether those factors were actually the ones that they considered in giving people steroids? Because I think it's this concept of who is the right patient to receive steroids. And if we don't know that, then any trial we develop is still going to be problematic.
Yeah, you're absolutely right. So to answer your question, we are going to be doing that. And we're also going to have a look at some of the blood-based inflammatory markers that are in this population that might, A, predict selection for steroid, and B, predict response to steroid. I think it's also worth pointing out that within a given site, there is likely to be variation from practitioner to practitioner. If you've got a CF clinic that has 3 or 4 practitioners, it's entirely possible that one of them might just be a steroid prescriber, and they might just believe in this steadfastly. And then other folks might say, I'm not going to prescribe steroid to anybody. And I'm also conscious that there may be some individuals in between, as evidenced by the sites where there's, say, 5% of patients who are prescribed steroid, who might say, well, you know what, most of the time we wouldn't do it for the first 19 of the 20, not indicated, but this person might have just something about them. They might just look that little bit hyper-inflammatory to me, clinical gestalt, and let's give them some steroid and give a rattle at that. What I would say is that approach appears not to work. So in terms of us using our bedside acumen to identify individuals who are good candidates for this therapy, I think we probably need something a little bit more objective and a bit more reliable.
Go ahead. Getting at that a little bit further, have you done subgroup analysis to look at those individuals who did seem to have a response, irrespective of what the bulk individual, when you aggregate the data, demonstrated? And could you use that then to be helpful in terms of finding biomarkers that might be useful to identify that population for future use?
Yeah, the answer is yes. There doesn't appear to be a clear rationale for why. I suspect that there will be an element of variation in terms of at what point in the course of their illness they were identified as having an exacerbation, and at what point in the course of their illness they were prescribed their initial antibiotics and steroid. And that's important. That's something that we've learned from COVID. When it came to looking at various, say, cytokine peaks and so forth, or shifts in inflammatory profile, the duration of symptoms and the time of onset is the key bit. When you don't account for that, your cytokine data in COVID becomes a scattergram. I suspect it's something similar here. I think the data that we generate from our biomarker study from the STOP2 cohort will answer a good deal of those questions. I think we will see some clearer signals emerge there, but I think it's a great question.
So there have been efforts with RCTs of steroids in exacerbations in CF, and that have met with challenges. And so what do you anticipate in terms of if we were to try and redo a steroid RCT, especially in the modulator era, and considering how much or how little we might know about what exacerbations look like now?
Yeah, for sure. I think that, again, we're borrowing and extrapolating from the more chronic disease trajectory when we talk about this, but there are data from our own group back in Dublin, as well as from North America and from Germany, that look at decreased inflammation in the airways of individuals with CF at baseline, and in particular at some of the inflammatory mediators, be they proteases like neutrophil elastase or master pro-inflammatory cytokines like IL-1 beta and IL-6, that are implicated in difficulty with pathogen clearance, with mucus hypersecretion and so forth. So I think that the face of exacerbations in CF post-modulator are going to be very different. That has implications for the power of studies. It has implications for the type of exacerbations we wanted to capture in clinical trials, and whether we shift more towards a symptom-based definition away from event-based in this kind of a study in future to capture more events opens up other problems as well, okay, in terms of the subjectivity of the data, how frequently we'll have to check in with folks to make sure we're really nailing the symptoms down. So there are challenges. I do suspect that now that the inflammatory profile of these individuals at baseline post-ETI, particularly for the F508del cohort, resemble that of non-CF bronchiectasis, I think we will be getting into the era of amalgamating those studies to increase power and then potentially get an indication for their use in both conditions.
Great, thank you so much. And we'll stop there.